Hi folks. So today I'm going to be talking to you about patterns of morphological and functional diversity across the early evolution, the origin. Across the origin and early evolution of a group called the acanthomorphs, which are the spiny-finned teleosts. So today, acanthomorphs represent a bewildering array of body plans. They're incredibly diverse, and they have a huge range of morphological and ecological specializations. So here you can see, for example, some puffers, a flatfish, a billfish, tuna, and an oarfish. However, surprisingly, the vast majority of this diversity was accumulated after the end of Cretaceous extinction, because they only appear first in fossil record in the Cenomanian. And the prevailing view is that they probably underwent this explosive diversification after the KT, as they filled this vacant ecospace that was left by the victims of the extinction. So this scenario of ecological release triggering a massive increase in disparity was quantitatively tested by Matt Friedman back in 2010 using a geometric morphometric study of 600 species of whole-body acanthomorph fossils, ranging in time from their first appearance in the Cenomanian, the early late Cretaceous, all the way through to the Miocene. And he supported this view of ecological release triggering this massive diversification. And he also was able to demonstrate that the diversification post-KT was, at least in part, to do with filling this vacant ecospace, left primarily by things like large-bodied predators that went extinct. Although this is quite a striking macroevolutionary pattern, the approach that was used is quite broad brush, and also it's not exactly clear what the precise relationship between morphological and ecological diversity is. So what does this pattern really mean in terms of ecological diversity? Does it mean anything? So So if you want to study patterns of functional diversification, there are a few better model systems than the acanthomorph skull. Modern ichthyologists working on modern fishes have developed a wide range of biomechanical models to accurately and precisely quantify feeding performance, functional performance, which have been extensively validated. So we know that these really represent functional performance. And they can also be applied to skeletal material, which is very important for fossils, of course. So I'll describe these in more detail in a bit. But here I just want to note that it's important that some of these measures can only be applied to three-dimensional material, not compression fossils like Matt used in his earlier study. However, this isn't so bad because compression fossils actually lose a significant proportion of the morphological shape variation that was present in life, as I've been able to show in some recent research that I've been doing. So the dominant axis of the dominant mode of shape variation in fully inflated fossils is actually associated with the width of the skull, and this is the axis that's lost in compression fossils. So if we want to accurately describe morphological variation, 3D fossils are very important. So for this talk, I'm going to be using the functional measures that I'll be describing in a moment and three-dimensionally preserved fossil fish crania from this key period in acanthomorph evolution to trace patterns of functional and morphological diversification, and look at the ecological interactions with more deep-branching teleosts. And I'm not going to spend so much time talking about this, but at the end I'm going to touch upon the relation between morphological and functional disparity. So looking at whether morphologically similar taxa are functionally similar, for example, and also whether levels of functional and morphological disparity are linked. So, as luck would have it, we happen to have two remarkable deposits here in the UK that span this key period in the evolution of acanthomorphs. The first is the English chalk, which dates to the early late Cretaceous, and secondly we have the London clay. And so together these two deposits provide essentially a natural laboratory which lets us study patterns of functional and morphological diversification. And so both deposits preserve fossil fish crania in exceptional three-dimensional preservation. And these are essentially directly comparable to the sorts of information that you can get out of a recent fish material, which means that we can apply these biomechanical models that have been developed for studying extant organisms. So here we have a representative sample of English chalk acanthomorphs. So at this point in time, this is very close to the origin of acanthomorphs, and these consist primarily of deep branching representatives that today are species poor and ecologically marginalized. And in order to test hypotheses associated with invasion of ecospace, we've also included a bunch of non-acanthomorphs, although I won't get to talk about that in this talk. Here we have, moving forward in time to the London clay, we can see that at this point in time, acanthomorphs are much more diverse and also much more morphologically disparate, at least that seems that way from eyeballing them. And here we have some examples of London clay non-acanthomorphs. So in order to collect our data, we've been making extensive use of CT scanning technology. So far we've scanned actually considerably more than 150 specimens from maybe 75 or 80 genera. And if you invest enough time and effort, as Mimi Beckett, a DPhil student at Oxford, has done, you can get incredible digital preparations and dissections of this material. However, in order to collect the data that I'm interested in, thankfully, I don't have to spend that much time processing the data. And so here we have the landmark scheme that I've applied. And the great thing about having CT data is that we don't have to, this CT data, is that we can actually place the landmarks directly onto the CT data in a visa. So it doesn't require a lot of time preparing the data in order to collect the landmarks. So the landmarks were collected in a visa, then they were imported into R and analysed using geomorph, and the functional measurements were also collected simultaneously with the landmark data, and the functional measurements are calculated from those coordinates. So I'll just run through some of these functional measures. First of all, simple jaw mechanical advantage. You probably all know about that, just the ratio of the in-lead to the out-lead to characterise whether a bite is adapted for power or speed. Then we have maxillary KT. This is a more complicated measure that involves modelling the highly kinetic elements of the acanthomorph feeding apparatus as a 4-bar linkage. And essentially this just shows that when the lower jaw opens, this drives the rotation of the maxilla, and it's this change in angle of the lower jaw to the change in angle of the maxilla that makes up the kinetic transmission coefficient value. And the interesting thing about this complex functional system is that it's characterized by many-to-one mapping, which means that you can get a whole bunch of different combinations of lengths to produce the same KT value, which is not the case for mechanical advantages. Another simple measure is a proxy for jaw protrusion. So many acanthomorph fishes have extensive protrusion of the upper jaw with the premaxilla here, and you can get a pretty good proxy for that by looking at the length of the ascending process. So I've done that. And lastly, we have suction index. So suction feeding is very important in many different types of fishes, not just acanthomorphs. And characterizing this system it basically brings together all of the previous functional measures that I've talked about. And so this metric called suction index has been developed by researchers at the University of Davis, University of California in Davis. And it essentially describes how the input force on the in-lever here from the pachycerebral musculature acts to increase the volume of the buccal cavity here, and that interacts with the size of the gate to produce varying levels of suction generation. And because this is a complex functional system, you're able to vary the individual components in different ways to produce the same value of suction index. So this chart here just shows how if you vary the input lever and the mouth diameter, you can converge on the same values, these isoplanes. Having said that, people have noted that there's often a general trend in the overall shape of the body from these sorts of more broad-headed, long-faced forms with large gapes, all the way up to the more deep-bodied, laterally compressed forms with small gapes, going from low to high suction feeding generation capacity. So moving on now to the results. Here we have a morphospace with the first two principal component axes, which summarize respectively the variation associated with the width of the skull in the first principal component, and basically the elongation of the skull on the second principal component. Here we have the non-acanthomorphs and the acanthomorphs, the distributions of the acanthomorphs and non-acanthomorphs. And if we look at the change in range from the Cretaceous for the acanthomorphs through to the Paleogene, you can see that there does seem to be an increase in the morphospace occupation during this time. We can also ordinate all of our functional traits using a principal coordinate analysis and produce a similar sort of a plot. So here the first principal coordinate is primarily correlated with the closing mechanical advantage, jaw closing mechanical advantage, whereas the second principal coordinate is associated with suction index and the opening mechanical advantage. And here are the morphospace, function space occupations of acanthomorphs and non-acanthomorphs. However, when we look at the change in occupation between the Cretaceous and the Paleogene for acanthomorphs, there doesn't seem to be that much of a difference. So if we look at multivariate variants, summarizing all of the principal coordinate axes, we actually see that, contrary to our expectations, we don't see a significant increase in functional disparity between the Cretaceous and the Paleogene, which really surprised us. And it doesn't really matter how you look at this on individual measures. The closing mechanical advantage, KT, suction index, in no case do you find higher functional disparity in the Paleogene acanthomorphs relative to the Cretaceous acanthomorphs. Although I should point out that none of these are statistically significant. However, when you look at the patterns of morphological disparity, completely at odds to what Matt Friedman found in his much larger study, there doesn't seem to be higher morphological disparity either. So I'm going to have to hurry through here. So just to finish off, I'd like to quickly show you a couple of the neat functional correlations between morphological and functional data. So the first one kind of echoes the pattern that's been described in living fish. So the first PC is significantly correlated with suction index. So the first PC is associated with the variation in the width of the skull. So that's nice. It seems to jive with what we see in extant taxa. And it belies the many-to-one mapping, which is supposed to decouple morphological and functional disparity. And again, a similar situation with KT and the third principal component, which is associated with the relative length of the lower jaw. And that's quite a significant, this is a phylogenetic adjusted R-squared of 0.46, so that's quite strong. So I'll just finish up with a couple of quick conclusions. First of all, based on the data that we currently have, functional disparity doesn't seem to be significantly higher in the Paleogene for acanthomorphs, which is quite surprising. It could be due to any number of factors associated with, for example, taxonomic sampling. Perhaps there are taphonomic effects going on with the deformation of the fossils. So this is something that we're going to investigate further. I wouldn't say that was our final conclusion. And secondly, just very quickly, it looks like there are all sorts of interesting correlations between gross morphology and a range of different functional measures, which actually bodes quite well for studies of morphological disparity, reflecting this ecological diversity. So with that, I'd like to thank my collaborators, the Leeds Trust and the Natural History Museum and these other museums, and thanks very much.